This is lecture 2, part 2. We're continuing with the lecture on blood, and we'll talk now about blood transfusions. Generally, whole blood transfusions are rarely done now. The blood is collected and then spun down, and the plasma typically is removed, and what's given to patients are packed red blood cells. Now, blood that's transfused is typed for blood groups, and the key thing that are tested for to match between donor and recipient are the ABO blood group and the Rh big D antigen. There are, in fact, the antigens structures on the or molecules on the surface of red cells are not restricted to those, but there are in fact many. However, the response to ABO antigens and big D antigen, if a person has been immunized with big D, then been exposed to it before, then those kind of reactions are very strong, so it's important to test individuals for those. As I said, the major blood group antigens are ABO and Rh big D particularly, which can cause strong vigorous transfusion reactions if given to an inappropriate recipient. And so we test for ABO, and when testing for ABO, what's being looked at is whether a person's red blood cells express the A antigen molecule on the surface or the B molecule on the surface of the red cells. Now, the unique thing about ABO blood group is that people who have not been exposed to A blood, and this of course is as long as it's not, they themselves are not A, you're always, all normal immune responses are against non-self. You never react to self. If you do, then that's an autoimmune response. So the key thing about immune reactions is that they are targeted against non-self antigen structures. An antigen in the end is defined as a molecule which can induce an immune response. So only non-self molecules will trigger immune response. And the unusual thing about the antigens associated with the ABO blood groups, A and B, those two antigens, is that they're really the exception compared to every other possible foreign molecule out there. I mean, there are billions and billions of foreign molecules that could potentially get into you. Some are associated with microorganisms, and that's how we defend ourselves. And some are on the surface of inanimate molecules that might get structures that might get inside you. But the key thing is that for every immune response, except for the A blood group and B blood group antigens, for every single immune response to any other structure, the billions of different non-self structure foreign target antigen structures that are out there, you have to be first exposed to it before you have a antibody response or immune response. However, and it's kind of unfortunate that students only really their first exposure to, in a lot of cases, to the immune system or immunity or antibodies is that they learn about blood groups, because these two antigens, A or B, are the exception, not the rule. So people who have B blood group will naturally and spontaneously have in their plasma antibodies against A. They will have anti-A in their plasma. And people who are the A blood group, in other words, they have the A antigen on their cells, will spontaneously, naturally have anti-B. Remember, the antibodies are just protein molecules that have been produced by particular B cells that can react to the antigen. But with the exception of those two, everything else, you are always need to be first exposed to it before you respond. And this is what this is a summary, I guess. You could call it a summary. I don't try not to use that term. It is used, but I try not to use it because it's sometimes for some students it's confusing. If a person is has the ABO blood group AB, then that means they have the A structure, the molecules on the surface of their red cells all over the surface. And if they are B, blood group B, then that means they have the B molecule on the surface. So AB blood group has both of these. B has only the B structure on its surface, here, shown here. And A's have the A molecule on the surface. And blood group O means that the person's red cells have neither A nor B. And these individuals, as you probably already know, and certainly many students have learned this before, those individuals have antibody. The person with B ABO blood group will have anti-A naturally, spontaneously developed in their plasma. A's have anti-B. O's have anti-A and also anti-B. Remember, always against a non-self. They have an immune reaction. They'll have an immune response against non-self. ABs, they don't have any antibodies in terms of A or B because that would react with self. So, when a person is being typed, their blood is being typed, the blood group of the cells is determined because their samples of the blood are combined in separate wells or separate tubes with commercial anti-A, which is always blue. I may have made a mistake on the lab in the lab lecture. Anti-A, commercial reagent anti-A is always blue and commercial anti-B is always yellow. So these antibodies are added to the person's red cells, and if you get agglutination, which means a cross-linking of the cells, clumping or agglutination is cross-linking of the cells with the antibody, because each antibody molecule, which are much smaller than is shown here, much smaller than red cells, but just for the purpose of the diagram, the artist has made them bigger than they actually are. But they do have two antigen binding sites, so they can cross-link red cells, and you get this kind of clumping, or properly called agglutination. And so a person who has A and B blood group antigens you'll see agglutination in both these wells, and therefore they are AB. A person who's B will have no agglutination with anti-A, but will have agglutination with anti-B, and therefore they are B, etc. A's respond here, but not here. That defines them as A. And you get no agglutination for an O because they have neither A nor B antigen. Okay, the other major blood group that's always checked, or the cells are always typed for to match between donor and recipient, is the Rh antigen, red cell antigen, called big D. All right. Now, there are a number of different Rh antigens, including big C, little c, big D, big E, and little e, but only big D is highly immunogenic. In other words, it would trigger a strong immune response, and therefore the next time the person gets exposed to it, they would have a strong reaction. So big D is always tested for, and a person who is big D positive, in other words, they have that antigen on their red cells, they are defined as being Rh big D positive, usually referred to as just Rh positive. Don't just say A positive, say A Rh positive or B Rh negative or B Rh positive, etc. You should always be clear in stating what the Rh group is, and that's Rh that you're talking about when positive or negative. The antibodies against Rh big D antigen are not spontaneously formed in an Rh negative person. Rh negative people have to be first exposed to the big D foreign or non-self antigen before they start to produce any significant quantities of antibody against it. And once that happens, after they've been exposed or immunized, then on second exposure, there would be a transfusion reaction, and the antibody they had formed would bind onto the cells, the Rh positive cells that were transfused into that person. So as I said before, and I remind you, the only two antigens out there, non-self antigens that people respond to spontaneously, they have or spontaneously produce antibody against without any prior exposure, are A and blood group, red blood cell group A and B, the antigens associated with those groups, A antigen and B antigen. Everything else, and there are many billions of other foreign non-self molecules, all of those, first the person has to be exposed to it, immunized, before they produce any significant immune response. Now there is an important disease process that you should be aware of, and I explain this actually in detail in Lab 2, and so I'm not going to go over it here in this video. It's in the Lab 2 video and explained there. It is now completely preventable as long as an Rh positive, big D positive pregnant woman receives RhoGAM. RhoGAM is an anti-big D antibody. You can read about it in more detail in the lab. Now, transfusion reactions do occur. The most important are when there's ABO antigens involved, antibodies against ABO, in other words, anti-A or anti-B. The person is getting mismatched blood that they have antibodies to, or if they have an antibody against the Rh big D antigen. These antigens, the reaction is quite strong, and often you can get quite a severe response. There are, as I said, many other glycoprotein antigens on the surface of cells, and you can see those in Lab 2. There are many, many listed, but the ones that are really important are AB and big D. Transfusion reactions occur if mismatched blood is infused. People who have received many transfusions, or females who've had multiple pregnancies, often do develop antibodies against the less common antigens that could trigger a response, less common, other types of glycoproteins on red cells. So, what's always tested is the recipient's plasma against the donor cells to see if there's any reaction in a tube. If there is an antibody against donor red blood cells in the recipient, then you can get a transfusion reaction, which could result in diminished oxygen carrying capacity or agglutination of cells so that they impede flow of blood in the smaller capillaries. You can get rupture of the red cells, lysis, hemolysis, so that you get free hemoglobin. And that actually is quite a big problem because free hemoglobin in larger amounts will precipitate in the kidneys and can even cause kidney failure or renal failure. There's always a consideration in terms of having an important aspect in terms of the cardiovascular system is always to have adequate volume. If the volume gets too low, then the pressure will drop. And so a person who has bled quite a bit can go into shock because their blood volume is too low. And so their mean arterial pressure is inadequate to perfuse the tissue. Even with, the fact that they've bled and they have less of a capacity to deliver oxygen, it's still important that they have enough adequate volume in their system so that the mean arterial pressure is enough and they can perfuse all of the organs. Often, if a person has had a large blood loss, the first thing they try to do is get the volume up so the pressure will increase. It is, after all, a closed system, so if you put volume into it, then pressure will rise. The simplest thing that might get put in is isotonic saline, but then they might also put in, if they haven't matched blood yet, they may put in a glucose solution, saline with glucose, or plasma. Plasma can be delivered more quickly than blood. Blood has to undergo cross-matching, which can take a little bit of time. But they try and expand the volume quickly, otherwise the person might go into shock. Plasma expanders have osmotic properties that can help increase fluid volume. Plasma expanders include human serum albumin or dextran, which is glucose, a solution of glucose, and that can pull some fluid from the tissues into the vasculature due to its osmotic properties. And so that's one way of volume expansion that is potentially used. Now, just to remind you in terms of that overview of where the blood goes and blood circulation, blood leaves the heart through the aorta and goes into the systemic circulation. The arteries will branch repeatedly off of the aorta until they get into smaller arteries and then arterioles and finally capillaries. And it's only at the capillary level that you get exchange of gas or nutrients or metabolic waste. So, and the capillaries are very, very small. The red cells are pretty much going through capillaries in single file, so you can imagine how small they are. The blood after the capillary bed, where the oxygen is released to the tissues and they take it up, and CO2 diffuses from the cells into the blood. The blood then goes to the venous side of the circulation, of the systemic circulation, and then back to the heart. And from there it gets pumped up to the lungs to release the CO2, pick up more O2, and then goes back to the left side of the heart and gets pumped out into the systemic circulation. The molecule in blood that is carrying oxygen is called hemoglobin. Hemoglobin molecule is inside the red cells, essentially not 100%, but to a large degree. The hemoglobin molecules are, the red cells are a bag of hemoglobin molecules. It's hemoglobin that carries oxygen. Each hemoglobin molecule can carry maximum or bind up, when it's fully saturated, will bind 4 molecules of O2. Now it's a unique relationship between oxygen and hemoglobin because it's a reversible process. Hemoglobin has 2 jobs. One is to take up oxygen and the other one is to release oxygen. Seemingly opposite roles, but carried out in a very actually elegant way, so that in a manner which is due to changes in affinity of the hemoglobin for oxygen. So the ability to bind to oxygen varies depending upon conditions. And the key determining factor, it's not the only determining factor, but one of the key determining factors for affinity of hemoglobin for oxygen is the surrounding partial pressure of oxygen in the surrounding area immediately around the red cell where the hemoglobin is. So the partial pressure of oxygen, or the PO2, in the lungs is quite high, and when the PO2 is high, hemoglobin has a high affinity for oxygen, it'll take it up. And the PO2 in the tissues is much lower. They're using up oxygen, so they have a lower PO2, and there, in that lower PO2 environment, hemoglobin will release some of that oxygen, not all of it, some of the oxygen that it took up in the lungs. And that's really the secret to the ability of the molecule to have those 2 roles. It actually changes in shape. This idea of changing affinities is called actually positive cooperativity. This graph is a oxygen hemoglobin dissociation curve. That's one name for it. You might also see a name. You might also see sometimes the oxygen association curve or hemoglobin association curve, but it's typically the oxygen hemoglobin dissociation curve. What this shows, and it's in a very important graph that you should completely understand and be quite comfortable with, what it shows is the partial pressure of oxygen measured in millimeters of mercury, but we don't always say the units. I'm not going to say the units every time. I'm not going to say millimeters of mercury every time. I'll just give a number, but those are the units. The partial pressure of oxygen is along this axis, and the degree of saturation in terms of percent, how much the molecules are saturated with oxygen, is shown here along this vertical axis. Now I want to point out to you, even though this goes up to 100 here for PO2, or is marked up to 100, it is not a percentage. It is a pressure reading, partial pressure reading for oxygen. At rest and at sea level elevation, the PO2 in the lungs, and therefore the PO2 in the alveolar blood in the lungs, the PO2 in the lungs, in the alveoli of the lungs, and the PO2 in the capillaries that surround those alveoli will be at 100, and this is an important point here, not 100, but 104. Okay, so it's just here. Now, most of these graphs always show it at 100, but it's 104. I don't know why they don't mark it more prominently, but it's going to be at 104. When you go to the tissue level, you find at rest that the tissue PO2 is 40 if the person is at rest. All right. Now I want you to realize that essentially what's happening is that when the blood arrives at the lungs and goes into the capillaries around those inside the lungs in these structures called alveoli, where you're getting the exchange of oxygen and carbon dioxide between the blood and the lung space, alveolar space. When it gets there, it takes up oxygen because it has a higher affinity and will be very close to actually 100%, not quite 100% saturation. You know, the curve is, this is an asymptote. It's approaching 100%. You can see even at 80, if the PO2 was 80, it's still very, almost fully saturated, which means that we, of course, can actually live fairly comfortably at a much higher elevation where the PO2 is lower, like 80. But in any case, you get full saturation pretty much at 104. Now, the vascular system is a transport system for most of its length. The blood takes up oxygen in the lungs, hemoglobin binds the oxygen, and then goes back to the left side of the heart into the left atrium, then down to the left ventricle, and then gets pumped up the aorta into the what's called the systemic circulation that's delivering blood to the rest of the body. But there is no gas exchange except if you're in a capillary. So there is no gas exchange until that blood gets to a capillary bed in the tissues. And then it gets exposed to this lower PO2 of 40. Okay? Right here. So for its entire length, except for the capillaries, there is no gas exchange. It's simply a delivery system. And then suddenly it gets exposed. And at that PO2 level, hemoglobin can carry this amount, can be saturated just to the approximately 75% level. It's at the close to 100%, 98, 99 at 104, but it will suddenly come into be exposed to, in the capillaries and the tissues, to a PO2 of 40, and so it essentially is going to drop 25% of its oxygen. That's at rest. Now you may see graphs that have a slightly different shape. You may see 70% instead of 75%, but I wouldn't worry about it. Again, it's going to vary sometimes with the source. But it's these numbers 104 and 40 are commonly used for the PO2 in the lungs and the PO2 in the tissues, at least at sea level and at rest. Okay. Remember, you do not read this graph as if the PO2 and the oxygen is getting released as it goes towards the capillaries. It doesn't. It happens very suddenly when it gets exposed to that PO2 of 40 in the capillary bed. Notice that if it drops 25, maybe 30 sometimes % of the oxygen that it's carrying, it's still carrying an awful lot more oxygen. It goes back to the heart, to the right side of the heart, through the venous system, goes up the lungs, gets resaturated with oxygen. But if the individual was carrying out a lot of exercise, then the tissue PO2 would drop. It wouldn't be at 40, it would be lower, because a person is being really active metabolically. Now we're talking about muscle tissue. The muscles are actively using energy and generating energy, and aerobic respiration generating energy requires oxygen, so the PO2 can be significantly lower. But the nice thing is, let's say it's 30. Let's say the PO2 is at 30. The nice thing is, of course, that the hemoglobin, if it gets exposed to a PO2 of 30, will dump more oxygen. How much more? This much more. Let's do it in another color. This much more. Right? We're on a steep slope of the curve. This slope is fairly steep here. There's a big change in the drop of the amount of oxygen that hemoglobin can carry with small changes in PO2, unlike what you get up here in this region of the heart, where, you know, it's going to be fully saturated, pretty much 97, 98, 99, even at a fairly low PO2. And that's also good. That means you can take up lots of oxygen even when the PO2 is lower, say lower than 104, even 80. And you can dump an awful lot more oxygen when the PO2 in your tissues drops, because the tissues, for example, muscle tissue is highly metabolically active and needs that oxygen. Here, what they're showing actually is, and it's unfortunate, it's kind of a different shape. I don't think they paid proper attention, but here in a normal adult, this value, this curve here is a bit misshapen. But what they're essentially trying to show, well, it's not, no, actually it's not so bad. It's just, it's a little sort of not really as high as it could be. Maybe up in this area, maybe they just wanted to separate them so they're more visible. But it really should be closer to, you know, between PO2 of 80 and 100, it should be real close to 100% saturation. But what they're trying to emphasize here is human maternal hemoglobin does not have the same oxygen hemoglobin dissociation characteristics as fetal hemoglobin. The reason is that you know, the fetus is receiving oxygen from the maternal circulation at the level of the placenta. That's where the maternal circulation comes very close. It doesn't mix directly, but comes very close to the fetal circulation. And the capillaries that are present, both fetal and maternal, in the placenta will allow for gas exchange, but you can't exchange gas between two hemoglobins that are the same. You won't get any exchange. The hemoglobin will hang on to it. But fetal hemoglobin is different from adult hemoglobin, the maternal hemoglobin. Fetal hemoglobin has a different profile. It will have a higher affinity at a PO2 of 40, for example. It has a higher affinity than the maternal. Let me just change colors. Than the maternal hemoglobin, and therefore actually can pull hemoglobin from the maternal circulation to the fetal circulation. The other thing they're showing here is that lambda hemoglobin also actually has an extremely high oxygen saturation, even at quite low PO2s. And the reason that makes physiological sense is because llamas live at quite high altitudes, and where the PO2 is much lower, and so they need to have pretty good saturation of their hemoglobin in the lungs, where when they're living at a high altitude, where the ambient PO2, I'm not sure what it is where they live at those altitudes, but it could easily be a PO2 of 60. Now, another thing that's shown here is another molecule, which I would say is of interest. It's myoglobin. Myoglobin is a molecule that's related to hemoglobin. The structure of hemoglobin is 4 polypeptide strands that have a heme group at the center, such that the 4 polypeptide strands that make up the entire molecule have therefore a total of 4 hemes, and therefore can take up 4 oxygens. Myoglobin is only 1 strand with 1 heme, therefore it takes up 1 molecule of oxygen. It does not have this kind of S-shaped curve. It has a curve like this, what's shown here. So this is a unregulated, unchanging kind of exponential curve where you get rapid uptake at the very low end until it starts to get saturated and then levels off, asymptotes off. That kind of exponential curve, as opposed to a sigmoid curve like you have with normal adult hemoglobin, indicates that myoglobin is not regulated. It does not change its shape, actually. It has very high affinity at low levels, and then once it starts to get saturated, well, of course it's going to stop being able to, in this region, it's going to stop being able to take up much more because it's getting saturated. Whereas hemoglobin changes its shape and changes its affinity depending upon what the PO2 is. At lower PO2s, it has a higher affinity. At a lower affinity, and at higher PO2s, it has a higher affinity. Well, myoglobin, in fact, just like blood, myoglobin is red, and that's why muscle, meat, is red when you buy. It's not because of blood in the muscle. That's not in the meat that you buy. It's not because of blood. It's because of the myoglobin. So that myoglobin actually is acting as a sort of a last resort reserve of oxygen. If muscle tissue gets really, really short on available oxygen, then the myoglobin can provide some. But, you know, the myoglobin will not release any of its oxygen until you get into really low levels of PO2. So that's a final kind of reservoir of oxygen that the muscle may have. Well, I told you that this graph is extremely important, this oxygen hemoglobin dissociation curve. It explains a great deal about the physiology of gas exchange and how that occurs and how it occurs appropriately given different conditions. Hemoglobin, in fact, the normal curve, the normal pH in the body and the blood is about 7.4, right? And so here we see again at PO2 of 40, which you have in the tissues, you're going to be about, let's draw it in here, you're going to be at about 70-75% saturated. Whereas up here in the lungs, where you have, and it's not 100, it's 104, you'll have full saturation up in that area. Now, there are a number of factors that affect the hemoglobin oxygen affinity. And one key factor is the concentration of hydrogen ions. I remind you that the concentration of hydrogen ions, in fact, is what determines pH. So what we have is when you have a high hydrogen concentration, the pH drops. And when you have a low hydrogen ion concentration, the pH rises. Now, with a lower pH, where would you expect to have a lower pH? Well, you would have normally a lower pH in metabolically active tissue. And the reason for that will become apparent later. I'll show you why. But with in an area where you have a higher partial pressure of carbon dioxide, PCO2 is higher, you will have a higher hydrogen ion concentration, and therefore you will have a lower pH. So this pH here, for example, of 7.2 would be the kind of pH you might have in active muscle. And what that does is it causes a shift in the curve to the right. It's always described as a shift to the right. It's actually a shift more to the lower right in the graph. And so what that means, in fact, is with a small shift, relatively small shift in pH that is associated with metabolic activity, you get a change in the affinity so that in fact instead of having the hemoglobin at PO2 of 40, instead of it dropping this much, it will be able, at the lower pH, it'll be able to drop that much oxygen, that much more. So instead of dropping this much to here, it will drop all the way down to here, or release all the way down to there. That's because of the lower pH. And in fact, with metabolically active tissues, you probably also have, and this is something to keep in mind, and this is, you have to understand how things are regulated so that they operate efficiently in the body. Now, in the case of metabolically active tissue, you probably also have a lower PO2 in the tissue because the oxygen is being used up faster. So even a small drop in PO2 down to here, combined with that shift in the curve to the right, means that what you end up with is a really quite significant, if we intersect here, you get quite a significant increase in the amount of oxygen that's dropped or released in the tissues. This is the amount of oxygen, increased amount of oxygen here that's dropped in association due to the change in pH, but there is also a change in the PO2 in that metabolically active tissue, so even more gets dropped or released from the hemoglobin. These are concepts that you should be well aware of. This kind of effect of pH is called the Bohr effect and was described many years ago by Bohr, so it's named after him. The effect of hydrogen ion concentration, in other words, a drop in pH on the affinity of hemoglobin for oxygen. There's a decrease. The other important aspect, and this is independent of the Bohr effect, is the level of a molecule found in red blood cells called 2,3-BPG, which stands for 2,3-biphosphoglyseric acid, or 2,3-biphosphoglycerate. 2,3-BPG changes rapidly in its level. Red cells they're never free of 2,3-BPG. It's always being created at higher to lower levels. If you go up in altitude where the PO2 is lower, it will increase. You'll get increased production of 2,3-BPG. If you exercise, there's increased amounts of 2,3-BPG. This is found in red cells. The key thing is that it also causes a shift in the affinity curve to the lower right, just like with the Bohr effect and shift in hydrogen ion. If hydrogen ion goes up, that you get a shift. That's the Bohr effect. If 2,3-BPG goes up, you get a shift. And the same is true also for temperature. Increasing temperature also causes a shift to the lower right. And that's the kind of a thing you get when tissue is very active. As you know, when you're exercising, you're building up heat. The reason is that you're converting carbohydrate molecules through a process of oxidation and aerobic respiration. You're converting them into you're converting the energy in the carbohydrates into ATP. But the change into ATP is not 100% efficient. Some of that energy conversion also goes to creating heat, so your temperature rises. But the body uses that physiologically. The body uses that as to its advantage because hemoglobin will shift. The oxygen hemoglobin dissociation curve will shift to the lower right. Same goes for 2,3-BPG. It increases with exercise, increases when the PO2 is lower, and of course the Bohr effect. All of them are contributing to increased ability to deliver, in other words, release more oxygen under those kind of conditions where you need it. Okay. I think I'll stop here. So this is lecture 2, part 2. We're stopping here, slide 54.